We'll now go on to our next speaker. I would like to welcome Talon Philippe Silva, who will be giving a presentation titled Hyperglycemic Environment Promotes M2 Polarization in Macrophages During Leishmania Infection and Impairs Antigen Presentation and T-cell Activation.
Hi, everybody. Can you see me?
Yes, we can see you. And if you're ready, you can share your slides and get started whenever you're ready.
Can you see my
we
presentation?
Can see your slides. Yes, please proceed.
Okay. So hi again, everybody. My name is Talon Philippe Silva. I am a PhD student in experimental pathology at State University of Londrina in Brazil. And I'll present my work entitled Hyperglycemic Environment Promotes M2 Polarization in Macrophages During Leishmania amazonensis Infection and Impairs Antigen Presentation and T-cell Activation. So I am changing the focus of the presentation a little bit. Everybody's talking about viruses, so I'm talking a little bit about Leishmania parasites. So the aim of this work was investigate if hyperglycemic environment influences the course of Leishmania amazonensis infection. And in order to do that, we did an in vitro model of, we did an in vitro model of hyperglycemia with 30 millimolar of the glucose, and we used THP1 cell line differentiated into macrophages with a low dose of PMA. We did these hyperglycemic stimulus for 14 days and infected the cells with Leishmania amazonensis parasites and evaluated the infection in several time points. We also culture cells in the same condition, but in a normal glycemic medium to use as a control in the experiments. First, we did an in vitro experimental infection using THP1 cells derived macrophages and L. amazonensis, and evaluated the parasite load inside the cells by real-time PCR, by quantitative PCR. And interestingly, we found that there is no difference in parasite load between these two groups in 2 hours after infection. And after these 2 hours, we washed the cells to remove any non-internalized parasite and analyzed the parasite load again in 24, 48, and 72 hours. And interestingly, we found that after 24 hours of infection and onwards, hyperglycemic cells showed twice the parasite load when compared to the control cells, indicating that although hyperglycemia does not alter the phagocytic capacity of the cells, which is evaluated in 2 hours, it impairs their ability to eliminate the parasites over time, which could be a susceptibility factor for leishmaniasis. And considering that the difference that we observed starts in 48 hours, we decided to continue the next experiments only at this time of analysis. And to confirm the data observed in the qPCR assay, we did a recovery of the we did a recovery assay where we recovered the parasites from the cells after 24 hours of infection and analyzed their proliferation over time to access the amount of viable parasites recovered from the cells. We also did a scanning electron microscopy to see the parasites inside the cells and see if there's any difference between the groups. And we found that the recovery of viable parasites was 3.5 greater in hyperglycemic group than in control group. We also could see here in this scanning electron microscopy that hyperglycemic cells is way more infected than the control cells, indicating once again that probably the hyperglycemic stimulus impairs the capacity of the cells to fight against infection. And since there is this difference in the capacity of killing the parasites over time in hyperglycemic stimulated cells, we evaluated if there's any difference in the generation of oxidative stress in this context, considering that oxidative stress is the main factor responsible for killing the parasite once they are phagocyted. So we quantified the amount of superoxide anion in the cells and also the total production of reactive oxygen species. And so just clarified here, we have two sets of columns. We have the first one that represent control group. The second one represent the hyperglycemic group. The same here. White columns represent uninfected cells, while black columns represent infected cells. Regarding the production of superoxide anion, we found that the hyperglycemic cells showed a higher basal level of superoxide anion than control. And probably these higher basal levels of superoxide anion is related to the low-grade inflammatory state that is classically triggered by hyperglycemia. And we also observed several papers has been described this phenomenon in diabetic patients. However, after infection, these levels of superoxide anions in hyperglycemic cells significantly reduced, while control cells managed to significantly increase the levels of these free radicals. In the same way, control group was also able to induce the production of reactive oxygen species after infection, which did not occur in control group. Indicating probably the inability of the hyperglycemic cells to fight against infection is a result from this lack of oxidative stress generation after infection. And considering that difference between groups about oxidative stress, we evaluated the enzymatic activity of two important free radical scavenger enzymes, that is superoxide dismutase, or SOD, and catalase. Superoxide dismutase presented similar enzymatic activity levels in control and in hyperglycemic levels before infection. It presented similar basal levels. However, after infection, control group reduced the enzymatic activity of SOD, while hyperglycemic cells increased the activity of this enzyme. Similarly, regarding catalase, a control group may keep the same levels of enzymatic activity even after infection, while hyperglycemic cells increase the enzymatic activity of catalase. As I already said, sodium catalase are two important scavenger, free radical scavenger enzymes, which block free radicals and impairs their action in pathogens, for example, and promoting the successful of infection, for example, in this particular case of Leishmania amazonensis infection. In the same way, we also evaluated the enzymatic activity of arginase. Arginase is a key enzyme in Leishmania's escape mechanisms, and also is responsible for the generation of polyamines that is important for Leishmania survival and proliferation. And several papers has been has been demonstrated that arginase is a huge susceptibility factor during Leishmaniasis. And we observed that the hyperglycemic cells significantly increased the enzymatic activity of arginase after infection, which did not occur in control group. Besides that, arginase is also an important M2 macrophage marker. And since we observed this huge increase in arginase activity in hyperglycemic group, we decided to investigate if the hyperglycemic stimulus alters other macrophage phenotype markers. So we evaluated 3 markers for MU1 macrophage, which is HLA-DR, CD80, and CD86. And we also evaluated 2 markers for M2 macrophages, once again, arginase and CD206. Here we have control, infected control, uninfected hyperglycemic cells, and infected hyperglycemic cells. We observed that control group increased the levels of HLA-DR, CD80, and CD86 after infection, which did not occur in hyperglycemic cells that maintained the same levels as they're presented before infection. In contrast, the opposite occurs in hyperglycemic cells. These cells increased the levels of arginase and CD206 after infection, which did not occur in control group. And so in general, we observed that both uninfected groups present similar levels of all markers. However, after infection, control group increased markers related to the MU1 phenotype, while hyperglycemic cells increased the markers related to the M2 phenotype. And we know that M2 phenotype has an anti-inflammatory phenotype, which are responsible mainly by tissue healing and controlling inflammation, while ME1 phenotype is more pro-inflammatory than M2 and is responsible for to internalize parasites and other pathogens and eliminate them. So this anti-inflammatory profile observed in hyperglycemic cells could also explain why hyperglycemic cells fail to eliminate the parasites over time. So considering that Leishmania amazonensis impaired the increase in MU1 markers of macrophages in hyperglycemic cells, we evaluated, and also considering that these markers is not only a macrophage marker, but they also related to antigenic presentation, HLA-DR, CD80, and CD86. Besides being an MU1 marker of phenotype, they are also receptors related to antigenic presentation. So to evaluate if these impairment in the inducing, if this failing induce the expression of these receptors in macrophages could also impairs antigenic presentation, we did an antigen presentation assay using peritoneal macrophages from healthy and diabetic mice. We infected them in vitro with Leishmania amazonensis for 48 48 hours. And next, we added autologous splenic lymphocytes labeled with CFSC to co-culture them and investigate if these macrophages infected with Leishmania are capable to present Leishmania antigens and activate these lymphocytes and induce their proliferation and the production of interferon gamma. So we measure the antigenic presentation by the T-cell proliferation through the geometric reduction in CFSC fluorescence intensity, and also by the production of interferon gamma. Just to clarify, we have here two sets of graphs. The first one is for the controls of the technique. The middle one is for cells from healthy animals, and the last one is for cells from diabetic mice. The same is here. White is uninfected, black is infected with viable Leishmania, and gray is infected with heat-killed Leishmania. So we found that macrophages from healthy animals induce the proliferation of lymphocytes, as well as induce the production of interferon gamma when we compare to the uninfected group. In contrast, macrophages from diabetic mice completely fail to induce lymphocyte proliferation, demonstrating similar results to the uninfected group. However, interestingly, this group managed to increase some interferon gamma production, even without inducing the proliferation of lymphocytes, but of course in a much less intense way than the healthy cells. On the other hand, the infection with heat-killed Leishmania amazonensis resulted in a threefold less exacerbated interferon gamma production. However, in this scenario, diabetic group was able to induce the T-cell proliferation, which could indicate that L. amazonensis can effectively inhibit the antigenic presentation in diabetic macrophages. So in summary, hyperglycemia increased host susceptibility to infection by Leishmania amazonensis, mainly by impairing the oxidative burst generation during infection, which prevents the cells from eliminating the parasites. And also in the same way, infection increases the expression of M2 macrophage markers in hyperglycemic cells, predisposing the cells to infection and impairing antigenic presentation capacity of macrophages. So here is some of my references. Thank you very much for your attention. If you have any questions, I am open to it.
Thank you very much for that presentation, Talon. We will have people be leaving questions in the Q&A section. So right now we do have one question. The first question is, have you looked at the M2 phenotype in mouse models? And have you looked at other markers of T-cell activation?
For phenotyping in peritoneal macrophages, we have looking and we have very similar results, but because of time, I didn't bring any of these results because this is only part of our work. So, but we have very similar results than we found in vitro with THP1 macrophages. And regarding T lymphocytes, we did not evaluate any other activation marker, only the reduction in the CFSC fluorescence and the production of interferon gamma.
Thank you very much. If anyone has any other questions, please post them in the Q&A section. And Talon, you can go to the question section and just type your answers to them.
Okay.
Dr. Fish, do you have a question?
I just, quick question, and again, I may have missed it, apologies. So humans, diabetics. I'm not familiar, I know with certain infections, but with Leishmania, do they have higher infection rates or worse outcomes? I guess it's worse outcomes that might.
Related to Leishmania infection or any other infections?
Well, Leishmania, let's talk. I know with other infections, viral Leishmania.
In Leishmania infection, this is a very, it's not new because diabetes and Leishmaniasis is not new diseases, but it's very, very little studied. So we have very few works that investigate this relationship. And so none of them investigated the mortality rate, for example, but there are some ones that investigated the morbidity of Leishmania in diabetic patients, and diabetic patients in general present abnormal lesions, skin lesions of Leishmania, with a difficult, with very difficult treatment. These lesions doesn't respond to traditional treatment, and the patient has to receive a lot of doses of glucanchime, for example, which is the mainly treatment for leishmaniasis, but it's very toxic, and in general, this patient doesn't progress to cure. And
thank you. All right.
I appreciate it.